Ajay Mistry, who is a chief executive at a company called Oppolotech. He has also established and managed a European investment portfolio for the major Japanese corporation Olympus, and has worked in various business and development roles for various companies such as Abzena and Synpromics. So take it away, Ajay.
Excellent, thanks. Yep, so I'd like to thank the organisers for the invitation to speak. Well, it's been an excellent conference so far. So I will start off by saying that us humans are very poor at making predictions, whether it's in economics. I mean, very few people predicted the stock market crash in 2008 and the banking crisis in 2008. Or in politics, very few people predicted Trump's victory in 2016. Or sports, I don't think anybody predicted Leicester were going to win the Premiership in the 2015-16 season, or pandemics. I don't need to say anything there. But another area where we get our predictions wrong is in the drug discovery process. And of course, getting predictions wrong here can cost hundreds of millions of pounds. And we often get our predictions wrong right at the start of the drug discovery process. In other words, at identifying and selecting the drug target. So we fundamentally believe that selecting a drug target is key to the success of the drug discovery process, and a lot of focus should be applied there. So this is a quote from Mene Pangalos, who's essentially the CSO of AstraZeneca, and he states that a selective high-quality molecule will never become a medicine if it is modulating the wrong target. This is why target selection is the most important decision we make in research. We completely comply by that statement. So why do we get our target selection wrong? We think there's two key reasons. First, we underestimate the complexity of cells. And secondly, we tend to view targets in isolation. So let's look at the complexity of cells issue. So if you look at any cell, it can be divided into these different layers. You've got DNA, RNA, protein, and metabolites. And life is an interaction of these different layers. But when it comes to drug discovery, especially antibacterial drug discovery, we've focused at the genomic layer. So what people have done is knocked out a gene, and if it kills the bacteria, they think whatever protein that gene encodes is a great target to go after, but not a single antibiotic has been developed that way. And we believe that we need to take more of a holistic look here. We need to look at all these different layers, the DNA, RNA, protein, metabolites layers, when you want to find a drug target. So the other reason we get our target selection wrong is that we tend to view targets in isolation. So this is a classic cancer pathway, which I'm sure you've all studied in your undergraduate years. It's the KRAS-RAF cancer pathway. And if you look at this pathway, the protein to drug is KRAS. That's what the data comes out with. But the problem with KRAS is structurally, it's a very smooth protein, so it's been very difficult to drug. And it's only now, some 20 years after the discovery of this pathway, that companies have finally cracked. In fact, Amgen have a phase 3 program targeting the inhibition with KRAS. But what we're saying is that we need to be able to look at KRAS in the context of the network that it's part of. And there might be non-intuitive drug targets, either to the top or the bottom, that are easier to drug and that still modulate that pathway. And identifying them would be a key to developing, finding good drug targets as well. So we've developed a platform based on systems biology and machine learning to model biological processes, and we're applying that model to towards building a whole cell model of E.coli. So what's important for a computer model is the input data. So what goes into our computer model? Well, things like gene transcription and translation rates of enzymes, catalytic rates of enzymes, messenger RNA half-life, any known regulatory mechanisms, enzyme concentration. So that's just a flavor of some of the things that go into the model. So what's important is the output. What do we want to get out of the computer model? So importantly, we want to identify viable drug targets, how sensitive those targets are there any known bypass mechanisms, can we discover any new regulatory mechanisms. So we focused on modeling E. coli, it's the actual K12 strain actually. So E. coli K12 has something like 4,225 genes, encodes more than 10.7 million different RNA species, has that many different protein and over 300 million different metabolites. So we initially focused on modeling the outer envelope of E. coli. And so this is the peptidoglycan model. So the dots represents enzyme substrates and metabolites involved in the biosynthesis of peptidoglycan. And you can see them dancing around. So this is the chemical reactions taking place. And these yellow dots are forming, which are the peptidoglycan dimers that get formed. And then they get flicked out into the periplasmic space. And we've gridded the E. coli because it's important to, what we find is that it's important where those reactions take place as well. So you can imagine what we can do with our model. We can push down on different parts of those components and see what effect it has on in this case, peptidoglycan biosynthesis. So we also focused on modeling LPS, which, as you know, is the component of the outer envelope, and this is the LPS pathway here, and this is the phospholipid pathway here. And I've circled the enzyme LPXC because it catalyzes a committed step, and it's an enzyme that's easy to assay as well. And because of those reasons, a load of companies have gone after it as a drug target. But what you find is that when you develop inhibitors against LPXC, and you generate resistant mutants, what's amazing is that instead of getting inhibitors, instead of getting mutations in the LPXC gene, you get mutations over here in the FABZ gene. And nobody could explain that for a long time, and our modeling was able to do that. So basically, LPS and the phospholipid pathway are made at strict ratios. And when you develop inhibitors against LPXC, you slow this pathway down, LPS. And so what the bacteria does with its billions of years of evolution is that it figures out a way of slowing this pathway down. And it does that by making mutations in FABZ. So if you put the mutated parameters into our model, you can see that this pathway is also slowed down. And so you get in viability again, because those ratios become viable again. So that gives you an insight into our modeling. So it tells us that, okay, LPXC is a good target, but there are at least two mechanisms that can allow it to be bypassed. So what we did was we used the model to find an alternative targets involved in the synthesis of LPS. So this top left panel here represents enzyme substrates and metabolites involved in making LPS. And then what we've done is we've stripped off all the other components except for LPS. So the red dots represents LPS. And then what we've done is we used the model to find a drug target, and we started to push down on it, inhibit that drug target. And you can see that the levels of LPS go down when we push down on this particular drug target. So this indicated that this drug target looked like a good one to go after. So what we did was that we worked with a CRO based here in Cambridge, and there was chemical structure available for this particular target, and they basically undertook a computational docking study. So they docked to about 1.4 million compounds, and at the end of the day, they came up with a list of around 1,000 molecules, and we tested those in our labs. And what we found was that none of the 1,000 molecules had direct acting activities, but we found five hits that had potentiation activities against with rifampin. So this indicated to us that the compounds were having some effect on the outer envelope, allowing rifampin to get into the bacteria and have an effect. So what we next did was we just started the hit to lead process. So now what we're doing is we're modifying those hits and improving trying to improve the activity. And what we found is that just with one round of a hit to lead campaign, we've now got direct acting activities. So we've now got direct acting MIC values. So you can see that this, just with one round, we're getting MICs in the range between 16 and 64 micrograms per mil. So this is with one round. So what we're doing next is we're undertaking a proper hit to lead campaign where we want to improve we want to modify those compounds, generate analogs, and improve the MICs. So we've got a great team that's undertaken this work. So John is our founder. He's an academic based up in Leeds, and he's the brains behind the model. So if you've got any questions, he's online. He can answer them. Ed is our microbiologist, so he's got extensive experience in undertaking microbiology screening work, and he's worked at groups like LGC and GR Micron and GSK as well. Tony is our med chem, so he directs our med chem efforts once we found a target. Rui is our computational biologist. He worked closely with John on our modeling work, and Cora is another one of our founders who's had extensive experience in the anti-infective space, and Keith is our chairman. So just to summarize, we're building a dynamic computational model of E.coli. We're using the model to identify viable non-intuitive drug targets, and then we're developing drugs against those targets by using med chem routes. And we're building a pipeline of first-in-class drugs based on those identified targets. And yeah, we're very open to collaborations around our model or our drug discovery programs. We've built an extensive isolate collection as well. And so we can undertake screening services as well at very competitive prices as well. So feel free to contact myself, Ed, or John if you've got any questions, and we're happy to answer questions as well. I think that's it. Thank you.
Brilliant. Thank you, AJ. Thanks for the talk there. It's always really interesting to see where people making models to try and really comprehensive models to try and completely kind of encapsulate everything that's going on in a particular system. It's quite fascinating stuff.